In this module of chapter Biotechnology, let us learn a technique, polymerase chain reaction or PCR. This technique was invented by Carey B. Mules in the year 1985. This is used for gene or DNA amplification. When we talk about amplification, it means increase in number. Suppose we find some DNA at a crime scene, maybe from blood smear, and we know DNA can be isolated only from WBCs. RBCs don't have nucleus, platelets don't have nucleus, and as compared to RBCs, the number of WBCs are very less. So whatever few WBCs we get, that is the only DNA or proof we have from that crime scene. And if we start using that DNA and if something goes wrong, the complete DNA or proof whatever we have is lost. So we want that DNA to be amplified to large number, say for few millions, so that even if something goes wrong, you still have the same DNA. The process PCR or polymerase chain reaction is completed in 3 steps. The first is known as denaturation. Denaturation is the separation of 2 strands of DNA. The DNA that we get is a double-stranded structure. Suppose if this end is 3' and 5', and the opposite strand will be again anti-parallel in polarity. So this end is 5 and the other one is 3'. So to begin with, we need to separate these strands so that on each strand new DNA can be formed. For this process, we can use hydrochloric acid dilute or dilute base, or we can use high temperature. Treating DNA at high temperature is a common method of denaturation. The temperature used is 90 to 94 degrees Celsius in this process. DNA has to be kept for a very short period of time at this high temperature because we want only the hydrogen bond between these strands to be broken. If this temperature is given for a longer period of time, even the phosphodiester bonds between the nucleotides of DNA will be damaged. So at the end of the process denaturation, DNA strands are separated. The polarity here is very important, 3' and 5', because in the next step, while talking about the primer attachment, polarity is very important. Step 2 in the process PCR is annealing, means hybridization. Now the DNA strands are separated. On the separated DNA strands, new strands to be hybridized. To synthesize a new DNA strand, there has to be primer which is required. Primer is a small fragment of RNA which would initiate DNA formation. And see, primer is attached at the 3' end of the parental strand. So 3' end, it's a strand of RNA, so definitely it also obeys the rule of complementarity. So it will be anti-parallel. If it get attached at the 3' end, the polarity of RNA will be 5' here and 3' at this end. The important point to be noted here is primer will always come at the 3' end of the parent strand and grows towards its 3' end or parent strand 5' end. Third step of PCR is extension. 3 important conditions required for the process is 1. Temperature 72 degrees Celsius, 2. Enzyme DNA polymerase, and 3. DNA nucleotides as raw materials. The DNA polymerase enzyme in our body won't work at this temperature. Our body temperature is 37 degrees Celsius and at 72 degrees Celsius, almost all our body enzymes will get denatured. So we need a thermostable enzyme for this step, and it is isolated from a thermophilus bacterium called as Thermus aquaticus. Thermus aquaticus. The polymerase enzyme extracted from Thermus aquaticus is Taq polymerase. Taq polymerase and the DNA nucleotide acts as a raw material for DNA formation. The nucleotides will be added to primer and new DNA strand will be formed. So already primer is attached to the 3' end and nucleotide will be added to the primer, and gradually new DNA strand will be formed from the 3' end of the parent strand to the both DNA fragments. So now we have two double-stranded DNA molecules. So every time we repeat the process, the number of DNA gets doubled. The process can be continued until we get 1,000,000 copies. So the three important steps of the process polymerase chain reaction or PCR are denaturation, annealing and extension.